All right, continuing here. So again, spend some time looking at this. It is not the easiest thing to grasp the first time you see this sort of thing, all right? Okay, let's look at family pedigrees. So Mendels Principles apply to the inheritance of many human traits. For example, the presence of freckles or no freckles, the widow's peak on the hairline or a straight hairline, or free earlobes or attached earlobes. These are the dominant traits and these are the recessive traits. So you can just, look at your family. Actually, it's best to look at a wider group of people. But, more people who are red-haired, lighter-skinned will have freckles versus no freckles. The widow's peak is more common than the straight line, and the free earlobes are more common than the attached earlobes, because the dominant traits would be expressed with the homozygous dominant genotype, but also the heterozygote. The recessive traits are only really seen with the homozygous recessive genotype, which is going to be less common. So these recessive traits, which you can see are the phenotypes, are also going to be less common, right? So how can we study human genetics? It's not necessarily ethical or legal to have human mating experiments, but we can examine a lineage, family lineage, which is what a pedigree is. So geneticists will analyze the results of matings that have already occurred and generate a family tree or a pedigree. So for example, the first generation would be established at any point in time, but if you have genetic information on your grandparents, you can consider them the first generation. And then look at the second generation, could be parents, uncles, and aunts. And then the third generation would be where you are, okay? So for example, looking at attached or free earlobes, notice that the free earlobes are going to be the open circles, okay? And the attached earlobes are going to be the colored in circles. And then male and female are going to be the circles or the squares respectively. So there's Betty, Grandma Betty, and she's a female, and she had free earlobes, and granddad Aaron, for example. So those are the genotypes of those two individuals that we happen to know what they are, heterozygote, heterozygote. The F for free earlobes is the dominant gene. So that's what gets expressed. That's the genotype. That's the phenotype based on the key, a free earlobed male, okay? And notice that granddad Cletus has attached earlobes, and that means he had to have been homozygous recessive, all right? Now, their children, one person had attached earlobes. So the only way that could happen is inheriting a second recessive allele from the grandma, who happened to be heterozygote, so she could actually donate or contribute a recessive allele into her gametes, okay? Notice that the only way you can have the attached earlobes in a generation is if the generation above at least was heterozygote, okay? So track this down, follow it through, and make sure you understand how the recessive characteristic can be expressed in this example only by being genetically genotype recessive, homozygous recessive. And then again, if you look at the ones that are the phenotype being dominant, the free earlobes, that's because the genotypes are either homozygous or homozygous dominant or heterozygous. Okay, so some human disorders are controlled by a single gene or allele, and they can be inherited as a dominant or recessive traits, right? They can be dominant or recessive genetic disorders. If they're recessive, they tend to be less likely. If they're dominant, they tend to be more common, okay? Why? Because if it's a dominant disorder, it will express itself both in the homozygous dominant individual, but also in the heterozygote. So you don't need to remember all these, but we'll just talk about a couple of them. So recessive disorders, for example, albinism or cystic fibrosis, sickle cell anemia, right? Dominant disorders, achondroplasia, which I'll talk about, dwarfism, Huntington disease, another one, okay? And then one type of Alzheimer's, for example. So most genetic disorders are recessive. That's because if they were actually dominant, they would result in more fatalities in individuals, and then they would tend to die out in a population. Not just the individuals, but the actual alleles that might be dominant. So individuals who have recessive alleles but appear phenotypically normal are referred to as carriers. So for example, a type of deafness is based on a recessive allele. So the person is deaf if they are homozygous recessive. If they are heterozygote, right, dominant and recessive allele, or homozygous dominant, they are normal in the sense that they can hear normally. However, if you are a heterozygote, you're considered a carrier because you're carrying genetically the recessive allele. So notice here, if two heterozygotes for the hearing disorder, that is recessive, mate, well, there is a 25% chance, just look at the Punnett square, of their offspring being deaf. Right? There's a 75% chance, right, 25 plus 25 plus 25, that the offspring are going to be normal in terms of hearing, but there's a 50% chance, 25 plus 25 here, 50% chance that the offspring will be carriers of the disorder. So using Mendels laws and a Punnett square, you can predict the fraction or the percentage of the affected offspring, but also the genotype of the offspring as well. So a number of disorders are caused by dominant alleles. So for example, achondroplasia is a form of dwarfism. It's a dominant genetic disorder, which means if you have the dominant allele, in this case a capital D, you will have the dwarfism appear on you physically, right, the phenotype. If you are normal, in the sense of not a dwarf based on this condition, then you have to be homozygous recessive, which most of the population actually is, okay? So notice the homozygous dominant genotype actually causes death of the embryo. Where are we? Oh, we don't have it here. Why? Because if the individual is homozygous dominant, it doesn't get past, it isn't born. The individual dies. So this is a dominant disorder that actually is fatal to the embryo if the individual is homozygous dominant. So the only survivable situation is going to be heterozygous, and those heterozygotes are going to be dwarfs. If they mate with a normal individual, since the condition is dominant, notice the probability, 50% of the offspring are going to have the condition phenotypically, the dwarfism, because 50% of the individuals are going to be genotypic heterozygotes. Because just follow how the individuals, the normal individual can have gametes with only recessive alleles, but the dwarf is going to have gametes with the dominant or the recessive allele. So just match the gametes together in your Punnett square or in a Punnett square. 50% will have the condition, the phenotype. 50% will have the genotype of heterozygotes. 50% will be normal, and 50% will have the homozygous recessive genotype. Okay? So again, that's how you use your Punnett square and be comfortable with being able to come up and determine the percentages or probabilities of what the offspring are going to be. All right? So the heterozygote individual with a single copy of the defective allele will have the disorder. Okay? So a person with achondroplasia will have a 50% chance of passing that condition on to their offspring. So another one, some dominant alleles of the lethal disorders are much less common than the lethal recessive alleles, because the dominant alleles will tend to lead to a higher percentage of the population dying, especially if they die before they reach adulthood, before they can mate and pass on that disorder to their offspring. So, for example, Huntington's disease is a lethal disease that causes degeneration of the central nervous system by middle age. That's where it begins, and the individuals will die a bit after that, right? They will not reach old age. But by middle age, most individuals will have had offspring. So if a person doesn't know that they actually have this condition when they're younger, but they reproduce, then notice the Punnett square here, half of the offspring will inherit the dominant allele, in this case H, capital H, for Huntington's disease. If the parent with Huntington's disease, then the dominant allele mates with the normal parent, normal individual that is homozygous recessive, normal, right? They don't have the allele. The defective allele here is the dominant one. Okay? And again, Punnett square shows us that 50% of the offspring will have the condition. Okay, there we go. And the only reason Huntington's disease actually exists in the human population still is because the onset of the condition doesn't happen until middle age. If the onset of Huntington disease happened in childhood, well, those individuals would die before they reached adulthood, and the alleles then would not pass on to future generations, and most likely Huntington's disease would cease to exist in the human population. All right, so genetic testing. So tests can be done to detect the presence of alleles during pregnancy. Okay. So, for example, amniocentesis is a test where they remove some of the amniotic fluid, right, during pregnancy, and that will be able to be used to test the genetics of the baby, because some skin cells will slough off the baby and will be in the amniotic fluid, and they can draw it off and they can test it for genetics of the baby, right? Because skin cells have nuclei, and nuclei have genetic material of the individual. That's one test. Something called chorionic villus sampling is they test the placental tissue and are able to determine the genetics of the baby individual. It's another genetic testing during pregnancy. There's newer genetic screenings that are available now that are actually more accurate and less intrusive, because there's always some danger of harming the embryo with amniocentesis and chorionic villus sampling. But the idea is to check the genetics of the baby, the fetus, and usually the sooner the better, so that the parents and the doctors have information about the potential harm that might come to the child based on certain genetic disorders or diseases, and then decisions can be made on healthcare and or even terminating the pregnancy, depending on the genetic disorder. Okay, so there are some variations to Mendels laws. So Mendels' two laws explain inheritance in terms of inherited genes according to some very simple rules of probability. Okay, so but Mendels' laws don't fully explain some of the complex patterns of genetic inheritance that we see in nature. For example, color. It's not always just a red and white flower. Sometimes there's a pink flower, and sometimes there's a flower that's kind of red and pink at the same time. So this isn't explained by simple dominant recessiveness of those alleles. There's other stuff going on. So the conditions that we've seen before with dominant and recessive inheritance, that's actually very simple. And in reality, things aren't quite so simple. So we'll get into a little more complex genetic rules now. And one is called incomplete dominance. We'll continue with this in the next section.